Alright, so start of a new topic. We're going to go through all of the different types of gene and chromosomal mutations in this lesson today. Before we do that, let's just go back through some key terms from last year in Year 11. So first of all, genotype. These are the alleles that you get from each parent that each organism has for a particular trait, so things like eye colour, hair colour. The phenotype is the expression of one of those alleles, okay, or in some cases actually both. So this is what we observe. This is the observable characteristic. So as we said up above, an allele is an alternate version of a gene, and a gene itself is a piece of DNA that codes for a particular protein. So how can we change the frequency of the alleles that we find in organisms? Well, the one that we're going to focus on today is mutations, and then in the coming lessons we're going to talk more about genetic drift, gene flow, and natural selection. So, mutations, what are they? A mutation is a permanent change, it can't be undone. And these permanent changes occur to the DNA sequence or to the chromosome structure themselves. So we have two different types here. We have changes to the DNA sequence, a gene mutation, and we have changes to chromosome structure or to the number of them, a chromosomal mutation. So mutations themselves can create new alleles that are going to code for these different proteins that we may or may not end up with. So how do we end up with mutations? Well, number one, there are mutagens, and mutagens are the things which can or are capable of causing mutations. There can be issues with DNA replication, and there can also be issues with cell division itself. So obviously the errors in DNA replication are going to relate to our gene mutations, and the errors during cell division are going to result in some of our chromosomal mutations. So mutagens, a factor in the environment that increases the frequency of mutations occurring. So we can have a few different types of these, the first being biological. So when we're looking at biological mutagens, we're thinking things like bacteria and viruses, as we can see down in the bottom right-hand corner. Secondly, we've got chemicals, so we've got carcinogens, so things like cigarettes. We've also got other nasty things found in foods and preservatives, some of the cleaning products around the house, even other chemicals like mustard gas and formaldehyde found elsewhere in the world. Finally, we've also got some physical mutagens, and these physical mutagens, the most common one is UV light. Second to that, really X-rays for medical use, but then also radioactive waste and nuclear explosions, but they're obviously quite limited examples. So all of these three examples of mutagens, so over on the left, physical, chemical, biological, cause this permanent change in the DNA known as a mutation. Now mutations can affect two different types of cells. So up the top, number one, they can affect a body cell, a somatic cell. And in this case, all of the daughter cells from that somatic cell will be affected. Okay, so if this occurs in a skin cell, when that skin cell goes through mitosis, all of the daughter cells arising from that original cell and its next generation, so on and so forth, will have that mutation present. Another case though, example number two of mutations, having quite a different impact this time, is a mutation in a gamete or in a germline cell, a sex cell. So in this case, the mutation will be passed on to offspring. Okay, so it's only in germline cells, if we have a mutation, that mutation will then be passed on to offspring. In the case of mutations in somatic cells, they will not be passed on. So what are some of the effects that mutagens can have in order to cause these mutations? Well, I've put a few things in bold here. Firstly, as we've mentioned previously, we can have errors in DNA replication. So what are some of these errors that we can have? We'll go into them in a little bit more detail in a second. Secondly though, we can also have DNA breakage and DNA extension, and we can also have damage to the DNA structure itself. Finally, we can also have mutagens reacting with and modifying the DNA itself. So those cells that have this damaged DNA in any way, shape or form will then multiply, and it's going to result in either a gene or a chromosomal mutation. And it will also then be either passed on or not passed on to offspring, depending on if it's somatic or germline. So when it comes to the mutations themselves and protein production, we already know, sure, DNA contains genes. All of them are coded for by particular amino acids, and those amino acids, genes code for the amino acids, I should say, and all of those amino acids are what determine our particular protein. Now, if we were to change a single base, it can potentially change that amino acid, and as a consequence, the actual protein itself. So if we change these proteins, you're probably thinking, okay, one really small protein won't have that big an impact, but we have a case study here of albinism. Now, albinism itself is only caused by one missing protein, okay? And that protein is responsible for providing the pigment to the hair, skin, and eyes. So some mutations, okay, will result in this mutation causing no protein. So down the bottom right-hand corner, we can see that a mutated gene is either going to produce no protein, aka in albinism, or B, it can cause an abnormal protein forming, so something that does a slightly different job perhaps. There is a third scenario though, okay? And some mutations can actually lead to the normal protein. There's no real change to the protein that's produced. So we have three potential options for every mutation here. So let's talk about how we can classify them. We've already mentioned a few things based on heritability so far. So we said somatic cells will be in, or somatic cells, mutations in them will affect all daughter cells. And we've said that all germline mutations are also capable of affecting offspring. Now, the key part we're introducing on this slide, however, is how there can be different mutations classified on their location. So our first is a gene mutation. This is a change to the nucleotide bases, and it's only going to affect one gene. Okay? On the other side of things, we have a chromosomal mutation, and this includes a change to the arrangement of genes, so it's more than one gene now, or to the number of chromosomes. So this is our heritability. Once again, I'm going to skip over it for now, but a nice image if you want to pause. Moving on to heritability, just a quick comparison. Again, I'm not going to go through this now, but if you want, feel free to pause. So when it comes to populations, and this is something that we'll get into much more heavily down the track, mutations can be really good for us because mutations introduce new alleles into the gene pool. Now, this provides more variation in a population, and it can, in some cases, help that population, help that species to survive in its environment, to combat a particular selective agent. Now, some mutations, on the other hand, in the not so good side, are harmful. Okay? And some mutations are going to result in the death or in the disadvantage of particular members of a species. Now, some mutations, okay, have absolutely no effect on the protein produced, and while there is a change in the DNA sequence itself, it doesn't have any lasting impact on the protein produced, as we mentioned previously. So in order for a mutation, and this is a really critical idea for us, in order for a mutation to have a lasting impact, we know that it must be a germline mutation. If it's not a germline mutation, it's not going to be passed on to offspring. So an individual could have a mutation of a somatic cell, so a body cell, and it could be really good for them. It could be advantageous for them in their environment. However, that won't be inherited by their offspring, and as a consequence, it won't have any effect on future generations. So let's talk gene mutations. Let's talk the three main examples for us. So when it comes to our gene mutations, the one that we're going to focus on at the start is substitution. As you can see, we have our original sequence at the top of TGGCAG, and what happens here is we simply substitute. We swap that C for another nucleotide. Okay? Now, what this causes is it may cause an issue with the amino acid that is produced by that triplet or that codon, and as a consequence, that can change the protein. or it could have absolutely no effect, and simply swapping the C for the T does not in fact change the amino acid that is coded for. In our next example, we have insertion. And in the case of insertion, we have quite literally taken this triplet and inserted it into the original sequence. So we still have TGG on one side, we still have CAG on the other side, but we've just introduced that TAT triplet in the middle. And again, this might add an extra amino acid, changing the protein, or it could add an extra amino acid doing absolutely nothing. Finally, we also have deletion mutations, another type of gene mutation. These deletion mutations, as we can see, remove nucleotides from the sequence. So instead of having the original 6 nucleotides, we're now cut back to 4. Okay? So gene mutations themselves are only affecting one gene, or a really small section of the DNA. Okay? It doesn't impact the entire chromosome itself. And it can be due to either the substitution, the insertion, or the deletion of particular nucleotide bases. Now, all of these can be an example of what we call a point mutation. Okay? And a point mutation is simply where we have one base, one nucleotide being replaced by another during DNA replication. It's only affecting that one thing. So again, I've highlighted this in yellow to replicate that substitution mutation, another name here being the point mutation. So ultimately, in the case of a point mutation, there can be no change to the codons any further down from that mutation. So again, while we've changed, if I highlight or circle in blue here, while we've changed this nucleotide, you'll notice that following that, the A and G have not changed. So it hasn't actually affected anything further down. So what are some of the effects of these substitution reactions? Well, number one, as we've sort of mentioned, this can result in a codon that ultimately provides the same amino acid, so no change. Secondly, if we get a different base, it could actually lead to a change in the amino acid. Okay? And finally, in some cases as well, it won't actually lead to the production of a different amino acid, it might just produce a stop codon. And that stop codon will lead to an incomplete protein, which can be quite bad in some cases. So this is another representation. So at the DNA level, we can see that originally we had a sequence of TTC. When we follow that down into the mRNA level, we have AAG. And at the protein level, this is going to code for lysine. Now, in our first instance, we have a substitution mutation where we've substituted our cytosine for thymine. This means that at an mRNA level, it no longer produces guanine, but another adenine. And fortunately, AAG and AAA both code for lysine, so there's been no change in this substitution reaction. However, in the case where we substitute A instead of T at the start, so looking at the second example now, this actually results in a stop codon. And UAG being the stop codon means that lysine is no longer produced. Finally, if substitution mutation occurs in the middle of this reaction, so TCC instead of TTC, this results in another guanine forming in the place of adenine, and this actually results in a different amino acid and a different protein being formed. So there are quite a few different reactions that we can have here for all of these substitution reactions, or mutations, sorry. So when it comes to insertions and deletions now, when we're talking insertion and deletion, I'll just double check my color coding from earlier, so pink and green. The insertions and the deletions are both considered what we call a frame shift mutation, so I'll represent them here on with blue. Okay? And what this means is rather, while a substitution reaction only affects a particular nucleotide, frame shift mutations affect everything further down, everything downstream of that initial mutation. Now, the reason being is that these mutations actually change how many nucleotides occur in a particular sequence. So if we look at this case here, in the case of deletion down the bottom left, in the case of deletion, we remove one nucleotide from the sequence and it causes everything else, if I highlight in blue, to move up one slot. And this changes the order that these amino acids are read. So if we're reading them as triplets, previously it would have been TAA, CTG, CAG, and then GT, so on. Now, however, following the removal, it is now going to read TAA, CGC, so a difference here, AGG, another difference here, and then T, so on. So we're getting a difference for everything following. Up next, as we can also see with the insertion over on the bottom right-hand corner, we can see that originally same sequence, so TAA, CTG, CAG, and GT onwards. Now we have TAA, much the same. However, the introduction of the C nucleotide has meant that we now have CCT, which has changed, GCA, which has changed, and GGT. GGT has been a change as well. So ultimately, these frameshift mutations have a much larger possible impact than a point mutation. So here are some quick review questions. Again, feel free to pause and have a go at some of these, but for now, what we're going to jump into is part 2. We're going to look at some chromosomal mutations. So when we're talking chromosomal mutations, as we mentioned in the earlier part of this lesson, they affect more than one gene, or they affect part of a chromosome, or they can affect an entire chromosome. Now we have 5 key types of different chromosomal mutations, being deletion, duplication, inversion, translocation, and finally, we can also have non-disjunction during cellular division as well. So let's talk a little bit about each of these. So in the case of deletion, simply put, a part of the chromosome is lost. Okay? Part of that chromosome is deleted. In our diagram here, we can see that part B, so this particular gene on this particular chromosome, is lost. Now, bad news with deletion, these ones are usually fatal, or they're associated with quite nasty conditions as several genes are contained on that portion of the DNA itself. Up next, duplication. In this case, instead of removing B, we now have 2 copies of that particular set of genes. And in this case, it can be harmful. However, it could also be beneficial in some particular instances as well. So all that we're doing with duplication is we're repeating one particular section of that chromosome. Moving on down to inversion, this is where a piece of the chromosome itself is flipped, and this can be a little bit tricky to see. So if we have a look over on the left on this first chromosome, number 1, we go A, B, C, D, E, F. Now what you'll notice, instead of going A, we've gone from A, B, C, D, E, and then finally down to F. So a little bit all over the shop. Now, these effects generally pretty mild, because while the genes are flipped around, they're not added, we're not getting an extra copy, and they're also not lost, we're not losing them entirely. They're still present on that chromosome itself. And our last kind of main mutation anyway is our translocation. So these ones, again, similar to our previous ones when we're looking up at our inversion chromosomal mutations. With translocation, pieces are moved around, okay? And they're not moved around on the chromosome now, they're moved around from one chromosome to another. So this is the key difference between translocation and inversion, okay? Now, this results in a pretty unusual arrangement, and in some instances can lead to some pretty harmful effects, as the body is a little bit unsure where some of the genes have gone. Okay, so when we're having a look here, we can see originally we've got uppercase and lowercase on chromosome 1 and chromosome 2. Now these get swapped around a little bit, and you'll notice here the main ones for us are E, F, and G, and E and F have swapped places. Okay, so here's another quick summary showing these 4 types of mutations. But now let's move on into our last little bit, which is non-disjunction. So this is a quick recap of what's covered in year 11. But non-disjunction, in case you don't remember, is where during mitosis, a chromosome pair fails to separate. And this can result in a few different options or a few different circumstances. First one being where a daughter cell has an extra chromosome, so being N+1 or trisomy. And we can also have instances where a daughter cell ends up with one less chromosome, so N-1 or monosomy. So all up, this is known as aneuploidy, which is a change to that chromosome number. Okay. So when we have a look down here, here are our potential options depending on what happens. Over on the left is a normal meiotic division. In the middle, we have that failure to separate during meiosis 1. So if I highlight meiosis 1 in this colour here, you'll see that these chromosome pairs, this homologous chromosome pair here, has failed to separate, meaning that this cell is going to end up with slightly too many chromosomes, and this cell is going to be missing a set of chromosomes. As we look at our final example, again, we've got non-disjunction, this time, however, in meiosis 2. The impact here, again, failure to separate in one particular cell during meiosis 2, leading to trisomy or monosomy. So, an example here that we have a look at again in year 11, and I'll finish off here, is this is an example of trisomy. So this is where, again, there's been a failure to separate of chromosomes during meiosis. It could be either meiosis 1 or meiosis 2 in rare instances, and there is a third copy of the 21st chromosome. So this is another example of a mutation that can happen.